Welcome back to this next tutorial in our series on the changing brain. Today we're going to talk about the construction of neural circuits. And this topic will pertain to three of our core concepts in the field of neuroscience. Again, we're going to be talking about a different kind of neuronal communication. Communication that is mediated chemically, but in this session we're going to begin to introduce the concept of building synaptic connections, and that will enable both chemical and electrical communication to be possible in developing neural circuits. So we're going to see the genesis of this very important core concept in the field of neuroscience today as we consider the construction of circuitry. We're going to talk about circuits that are in large measure genetically determined, circuits that provide the foundation for behavior, such as the ability to detect potentially painful and damaging stimuli in our skin surfaces. And then lastly, we'll set the stage for a more rich discussion that I hope to have with you in our next tutorial about how life experiences can modify the structure and function of the central nervous system. And I'd like to introduce a hypothesis really that will allow us to understand how can chemical signals that have the ability to change the course of neuronal development be modulated by activity. And this hypothesis will then help us understand how life experience can change the structure and function of the brain. Okay, well, let's consider the learning objectives that I have for you in this tutorial. I want you to be able to understand what is a principal organelle that is responsible for pathfinding in developing axons, as well as the growth of dendritic arbors. And this structure is called a growth cone, so I want you to be able to characterize the structure and function of growth cones. I want you to be able to discuss the major classes of signaling molecules that guide axonal growth in the developing brain. And I want you to be able to have a cogent discussion of neurotrophins, and I want you to be able to discuss the mechanisms of neural trophin signaling. Well, hopefully all of these topics will make sense to you after spending a little time with me in this tutorial. So let's jump right into it. Well, I would remind you of our broad framework for understanding the forces that influence the developing brain. We talked quite a bit in our last tutorial about the concept of nature in brain development. That is, the lineage-derived signals that result from the expression of particular genes. And so this influence in brain development can be considered genetic specification. Well, today we're going to talk about the construction of neural circuits, which begins to engage our two other major sets of factors that influence the final shape and structure and function of neural circuits. Well, if indeed there is ever a final shape to them. What I have in mind is self-organization. Self-organization is mediated via activity-based mechanisms that play out as cells acquire the capacity to generate electrical signals. And that begins to shape the expression of a variety of phenotypes that circuits begin to acquire as they build out their connections. Well, as the developing embryo, fetus, and into postnatal life, developing infant begins to interact with the environment, then sensory motor experience becomes an important shaper of circuitry in an ongoing phase of circuit construction that continues into postnatal life. So the story of the construction of neural circuits begins in utero. It begins at a relatively later stage in embryonic development in the third trimester, but it will continue well into postnatal life. So many of the mechanisms we'll be talking about today reflect nature, that is genetic specification. However, we will begin to describe some of those factors that play an important role in mediating the influence of nurture. And again, that's going to be more primarily the topic of the next tutorial in this series. Okay, well, with that as a broad introduction, let's talk about some of these mechanisms that pertain to genetic specification and self-organization in early brain development. Mechanisms that will shape the growth and the viability of neurons and their connections in the developing central nervous system. Well, the first set of mechanisms that play out establish polarity in the developing neuroblast. Now, you'll recall that the neuroblast is derived from an epithelium, and all epithelial cells acquire a polarity. That is, there's an apical surface and a basal surface to their cells. And this process plays out in developing neuroblasts, although their apical and basal surfaces are increasingly difficult to recognize because increasingly these cells no longer resemble the columnar epithelium that first established the neuroectoderm. Nevertheless, there is the establishment of polarity such that from the apical surface of the developing neuroblast, an axon will emerge, and from a basal surface, dendritic branches will begin to grow out. Now, initially, with the outgrowth of these processes, we really can't tell whether they are destined to become axon or dendrite. We simply call them neurites. But as development progresses, a single axon becomes recognizable and differentiated from the apical aspect of the neuroblast. And that axon begins to acquire a distinctive organelle at the growing tip of that axon. That's the structure that we call the growth cone. And let's spend some time together thinking about the growth cone and what makes it such an important structure in the finding of axonal pathways in the developing brain. Well, if one were to look at the tip of an axon that is emerging, let's say, from a cell body somewhere in this direction, one would find that there is a large flattened structure that begins to develop as that axon begins to differentiate from just a neurite that might be extending away from this neuroblast. So this structure is what we call the growth cone, and it's characterized by a fairly flat laminar type of appearance. We call this a lamellipodium or a sheet foot. That's one way to understand that term. And then from that lamellipodium extend these long finger-like processes called filopodia. Now, what we're seeing in this figure is an illustration zooming in just on one portion of the growth cone, looking at a filopodium. And this allows us to understand just a little bit about the dynamics that shape this growth cone as it interacts with the environment of the developing embryo. So what we find in this lamellipodium is a rich supply of mitochondria to supply the energy that's necessary for the activity of the growth cone. We also find a variety of filaments. We find actin filaments that assemble in the filopodium, and this allows that filopodium to have a highly dynamic behavior as actin polymers are assembled from the monomeric elements. So the assembly of those filaments is responsible for the protrusion of a filopodium, and then the disassembly of those filaments allows for the retraction of a filopodium. Meanwhile, microtubules are being assembled and disassembled in the lamellipodium. So all of the machinery that's necessary for the construction and deconstruction of these filaments needs to be synthesized and then localized to this growth cone. Now, these processes are responding to signals in the environment through which this growth cone is exploring. And those signals are transduced via a variety of receptors that are present within the leading edge of this growth cone. So, for example, one can find a variety of channels, including our transient receptor potential channels that can mediate the interaction of this growth cone with chemical signals in the environment. There are other means by which calcium can enter this lamellipodium, and as you know, calcium is a very important second messenger system within all cells, and in particular within developing neurons. So calcium becomes a key mediator of the events that are detected at the plasma membrane of the growth cone, which then are transduced into changes in the structure and growth patterns of this growth cone. So this entire growth cone is a highly modal structure. So this growth cone can explore the environment within which this axon might be proceeding. It can decide that it needs to change directions, perhaps retract from growth in one direction and begin to extend growth in a different direction, perhaps an orthogonal direction to the original pattern of growth. So there is a tremendous amount of dynamism associated with this growth cone as it actively explores the environment, responding to chemical signals. And sometimes there is an important point of decision, is there not? There are what we might call forks in the road for a developing axon that's pioneering a pathway. We think of these forks in the road in particular at the midline of the developing embryo, where a variety of axonal projections must make a choice to cross the midline or not to cross the midline. Well, I thought this might be a helpful point to insert a study question to challenge you to review some of what you know about anatomical pathways, mainly those that you learned in Unit 3 of the course. So let's just pause for a moment here and take on the following study question.